Okay.
Hey, Dr. Brum. So thanks so much for joining me. Could you talk a little bit about what your position is now and yeah, what you're working on or anything
Yeah.
else you want?
So I'm an assistant professor in the Department of Oceanography and Coastal Sciences at Louisiana State University.
Cool.
So I'm in Baton Rouge, Louisiana.
Gotcha. And I know you work on marine phages, but can you talk a little bit about what your work is or some of your recent projects?
Yeah. So I study marine viruses, and if you guys do they know anything about marine viruses yet, or anything oceanography related?
So it's kind of cool. So my department is the Department of Environmental and Forest Biology. That's our
Okay.
that's our department name. But some of the students do have marine science minors or majors. So some students will have taken like a limnology oceanography kind of combined
Okay.
course. But
Okay.
this in my class is just like a general evolution course. So
Yeah.
I've mentioned that viruses exist, but I haven't really talked a lot about the ecological context for marine phages in particular.
Okay. Great, thanks.
Yeah.
Okay, so a good place to start is if your students have ever been swimming in the ocean or swimming in a lake or a river or anything like that, you get a little bit of water in your mouth, and every mouthful of that water has about 200 million viruses in it. So that can kind of freak people out, especially right now when we have a pandemic going on.
Yeah.
But the thing is, those viruses are not there, they're not infecting you. They are infecting the 20 million bacteria that's in that same mouthful of seawater. So those bacteria they're basically running our planet. Bacteria are everywhere. They're in charge of the carbon cycles, they're in charge of the nutrient cycles, and the viruses are there to kill, infect, and usually kill them.
Yeah.
So all of those viruses are evolved to hopefully perfectly kill their host.
That's pretty wild. So I work on ants and land snails a little bit, and there's kind of a famous ant conservation biologist guy named E.O. Wilson who has said that ants are the little things that run the world, and that gets quoted all the time in ant literature. In sort of people
Yeah.
writing scientific articles about the ecological importance of ants. But it's very humbling to be exposed to the world of especially marine phages and like how important they can be for biogeochemical cycling in the
Yeah.
majority of the world. Yeah.
I usually say microbes rule the world, but viruses rule
Rule the
microbes.
microbes. Yeah,
Yes.
yeah, yeah. That's wild. So specifically, what are some of the consequences of all this kind of bacterial death?
So a bacterium is basically just, a sack of organic matter, right? And so when a virus kills that bacterium, it generally lyses that bacterium. So that organic matter is now free. It's recycled by the other bacteria. So the
Mhm.
viruses kind of keep this recycling going at the very base of the food web, in the oceans as well as everywhere else. Um, Another consequence of that is that, of course, there's going to be the arms race going on. So bacteria don't want to get killed by viruses. And of
No,
course, I'm giving them personalities, but this is
it's
all
totally fine.
selection. Yeah,
Yeah,
I like to do it.
it's
It's so the bacteria don't want to get infected by the viruses. So there will be selection for the variations of the bacteria that can't get killed by the viruses, can't get infected by them. But at the same time, there will be selection for the viruses that can get around those host defenses. So some of you may have heard of CRISPRs, especially in gene editing. That is actually a defense mechanism by bacteria to be able to recognize viral DNA and chop it up.
Yeah, so one of the tricky things about gene editing is that before we could only do it on sort of model organisms, right? Because we
Mhm.
needed to know exact sequences and we needed to really specify which sequences we were developing the molecular tools to do gene editing with. But CRISPR-Cas9, that system has really like opened up a lot of potential for us to do gene editing on like non-model systems where we might not know as much
Yeah.
about the genome.
And there's even one paper showing that a virus has stolen the CRISPR system from the bacterial host.
What?
I know. It's Vibrio cholerae phage. I can't
Wow.
come up with the author's name off the top of my head,
That's
but
okay.
yeah, as soon as the host develops something as a defense, the virus is going to find a way to get around it.
Jeez, that's pretty wild. What do you say when people ask you if viruses are alive? I actually don't know your answer to this question.
My standard answer is you have to buy me a beer in order to discuss this, because it's one of those like
Oh, boo. Yeah,
it's one of
yeah,
those hand wavy things, you know?
yeah.
I mean, the strict rules are,
Mm-hmm.
it has to have ribosomes. That's the thing that separates them from other intracellular parasites,
Yeah,
right? There are tons of obligate parasites. Well, viruses don't have ribosomes, and that seems to be where we've decided to draw the line.
right.
In my opinion, I call them the undead, because I mean they're not living outside of a host cell. There's only one virus I know of that can actually change its shape outside of a host cell, which is pretty cool in
That is very
itself.
cool.
But 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 on the other hand, as soon as they get into the host cell, they just completely take over.
Yeah,
And so now people call that like one organism, right? That infected bacterium is now something completely different.
right.
So I say they're not alive, but
Yeah, yeah,
they're close. But
yeah,
it
and
doesn't change the function of what they do, is why I think this is like
sure.
a really hand wavy thing. It's
Yeah, and just from a strict, kind of, understanding evolution perspective, they dip in and out of the tree of life, so you can't really you can't really understand the full history of, the full evolutionary history of living organisms without accounting for some viral DNA and, without accounting for evolutionary arms races with viruses and things like that. So,
Yeah, so one of my projects is actually studying viruses and hydrothermal vent systems. And
oh,
so I've been
cool.
reading a lot about, like, there are theories on how viruses evolved.
Yeah.
As in, the theory is first, they were a means of transferring DNA between protocells. They were basically just carriers. It
Huh.
wasn't until later that they evolved to be, as I put it, murdery. Um, and then what that resulted in was actually pushing the evolution of these protocells, these first cells on the planet, because now they had to defend themselves against these viruses that had
Huh.
turned from delivery systems to things that might kill them.
Interesting. Wow. So that's kind of maybe the first example of like a parasitism evolving from a mutualism or something like that, like a breakdown of a
Yeah,
mutualism or something.
it's thought that the first viruses were in fact the ones that were temperate viruses that could integrate into their host genome. So it was
Wow.
like one cell has this one type of gene, it
Hmm.
can actually package that gene and go to another cell, and now that new cell has a totally new set of genes in it, right?
Yeah.
It's
Yeah.
quicker evolution than waiting for mutations.
Yeah. It's almost like community assembly at a certain point. Like when genomes were kind of more labile, sort of in the hypothetical deep past, if if there was maybe even more genomic recombination or more horizontal gene transfer happening. Like, are you? I don't know if this still has any credence, but the Carl Woese, I think, had like a term called like evolutionary temperature or something. He talks about. Yeah, never mind. I'm not prepared to talk about it. So
I'm not
yeah,
familiar
forget
with
about it. Yeah, yeah.
it.
How can we maybe back up and talk about you for a second? Because you didn't always want to study viruses, right? You didn't come out. Were you?
Well,
How did you
okay,
get into viruses?
let's see. You're right. I didn't first want to study viruses because I decided I wanted to be an oceanographer when I was in third grade. I learned the term oceanography, and of course, I was in
Nice.
third grade, so we were learning about whales, and so I wanted to study whales.
Obviously.
But then, obviously, because they're cool.
Yeah.
Then in high school, I found out about viruses and I got really into viruses. But this was before Google. That's how old I am. And all the books I could find about viruses were about viruses that infect humans.
Mhm.
And I did not want to study humans, just no way.
Yeah.
So I went to college to be a marine scientist. I went to Hawaii. And my sophomore year, my marine ecology professor said, "Oh yeah, by the way, there are tons of bacteria and viruses in the ocean." And I was like, wait, what?
Wait,
So
hold on a sec.
yeah, by that time I had learned, like, I was learning how to look up scientific papers, again, pre-Google.
Yeah,
And I just started reading every single thing I could find about them. And that was it. That's what I wanted to do.
that's awesome. That's really cool. And what's it, because you've been on some research voyages, right? What's that like? What's it like being on a boat for a super long time?
Yeah, so I've been on 18 research cruises, and I've also spent a couple months at a research base in Antarctica.
That's awesome.
Um, yeah, both times I've been to Antarctica have been the best experience of my life.
That's so
So
cool.
if you ever get a chance to go, just drop everything and go. But actually, my first research cruise was the summer after my junior year of college. It was only a 5-day research cruise. It's really weird because you're out there and you're just not getting much sleep. You pull a
Yeah,
12-hour shift, 12 hours straight, doing, collecting water samples, processing water samples, getting everything that you need. And then with the other 12 hours, you are supposed to do all your own research, right? All the extra research aside from what's needed on the cruise. So you get very little sleep, but I loved it. The first time I went out to sea, I was like, this is it. This is what I want to do. Microbes at sea.
that's awesome. Wait, but how much research can you actually get accomplished on the, because it's mostly collecting and kind of like early processing
Mm-hmm.
stuff that you can actually do, because a lot of your stuff involves either sequencing or electron microscopy
Yeah.
or
So we're not like physical oceanographers who can go out and just collect the data and then
yeah,
they can process it right there.
right.
When I go out to sea or I go into the field, I'm just collecting samples. So like the last research cruise I was on was a 5-week research cruise to hydrothermal vents on the Mid-Atlantic Ridge.
Oh, that's awesome.
And
Wait, when
yeah,
that must have been
fortunately,
a
I
couple of
was
years
on a
ago, right?
German research cruise. It was a German research ship. So that means, hi students, sorry, that means we got beer. Um,
I was
you
wondering
can't,
where you were going with, yeah, fortunately. Yeah, I feel like Europeans in general are much more likely to
yeah,
have
on
alcohol
US ships, you
there.
cannot have any
That's so
alcohol except
funny.
to preserve your samples.
Yeah.
But
Oh,
for that 5 weeks, I processed 1,000 liters of water.
oh my
That
gosh.
is actually my personal record.
That's a ton, right? Is that a metric? Is that a metric ton?
Sorry.
Processed a ton of water.
Yeah. So, but of course, you're taking small samples, you're
Right.
preserving them and putting them in the freezer. That's for doing like virus counts or bacterial counts. But for DNA, we like to get at least 20 liters of water per sample. And so we have a method where we precipitate the viruses. So we
Mhm.
add in some iron and that causes the proteins to glom together.
Hmm.
And then we filter it onto a filter. It's about that big. Then we
Yeah,
fold the filter up, put it in a tube, and keep going.
love it.
So 1,000 liters worth of water ends up, a couple of boxes like this. That's it.
Jeez. So, yeah, do you have time to talk a little bit about like some research that you're doing right now, or I guess the hydrothermal vent stuff is kind of ongoing as well,
Yeah,
or
that's not my primary project. I'm not
that's kind of
actually
more like a
funded for the
review.
hydrothermal vents one
Okay.
yet. I came close.
Uh,
But I am funded to do this study of viruses in oxygen minimum zones.
oh, cool. We
Yeah, so
actually
your
have
student?
Sorry, go ahead.
Oh, go ahead, please.
Oh, well, we have one of the faculty at ESF, Karin Lindberg, who works on fish otoliths. She's really interested in anoxia in the world's oceans too. She's kind of active in that research program. So that's something that some of our students may have actually heard a tiny bit about.
Yeah, I was about to say a lot of people hear them as dead zones. Like especially in Louisiana, we have a seasonal hypoxic zone, which everyone just calls the dead zone. But that's a misnomer, of course, because when you take away oxygen, microbes rule. I mean, they rule anyway, but they
there's plenty of things. Yeah.
So there's all sorts of cool biogeochemical cycling going on in these, and that's what I'm into. Is I want to know what types of viruses are found in these areas and how they're affecting the metabolism of their hosts and how that affects biogeochemical cycles. For example, oxygen minimum zones are a big source of nitrous oxide to the atmosphere. So that's a greenhouse gas.
Oh, wow. I
also
never made that connection.
Yeah, they're also a sink for nitrogen. And these things are huge. They're massively huge. The one off the coast of Louisiana is very small and it's seasonal, but there are permanent, huge anoxic zones.
Wow. So, and where do anoxic bacteria or where do anaerobic bacteria come from in the ocean, right? Because are they just like already there and they just kind of appear?
Well, there's a bunch of places you can get anoxia that you're probably not thinking of. One is if you go beneath the upper portion of the sediments at the bottom of the ocean, that'll be anoxic. But then also there's sinking particles. And there was this cool study done, I think it came out when I was a grad student, but they stuck a tiny little oxygen sensor into the middle of these particles and they're anoxic in the middle.
Oh, that's nuts. I have seen stuff like that for soil particles too, right? There's a whole oxygen gradient just in like
exactly.
a tenth of a millimeter.
So there's all these small niches that we're thinking of, and then those bacteria, and then of course there's bacteria that can exist within a range of oxygen concentrations.
Sure, yeah, like
so they'll be selected for around these areas, right? Yeah.
Cool.
So of course what I found is that the viruses in the OMZs are different than the viruses when there's oxygen.
Makes sense.
that was our basic hypothesis. It's good to
good to falsify that null right off the bat. Yeah.
Well, I tell my students it might take you like 5 seconds to say something. It takes you years to actually prove it.
Yeah, for sure. Yeah.
We have samples from 7 different oxygen minimum zones.
Oh, cool.
So this is another thing. Our other part of our research, your students may not know about these things, but viruses can have the genes for metabolism. So there are viruses that have the genes for photosynthesis. Not all the genes for photosynthesis, just like 1 or 2 genes for photosynthesis. And the reason why they have these is because their hosts photosynthesize. And so when the virus infects their host, it has the genes for the rate limiting step of photosynthesis. And so it ramps up photosynthesis of the host to make the host make more stuff so the viruses can assemble more of them. Then it kills the host. But in the meantime, viruses are actually directly influencing carbon dioxide and oxygen concentration on our planet.
Wow.
So it turns out those were some of the first genes that were found that viruses have, but it turns out viruses can have genes for carbon metabolism. They can have genes for sulfur and nitrogen metabolism. They can have, I've even seen a virus with the genes for flagella, and
wait, what?
Without doing any work, I really have this hypothesis that like the virus infects the host and causes it to swim faster. I want to call it the zombie virus.
Oh my God. Yeah, that is like a specific premise of some zombie movies, right? You get like super speed or whatever. That's nuts.
They're awesome.
Whoa.
So the thing is, my second main hypothesis for this was that those viruses in that oxygen minimum zone were going to have all the genes for this cool biogeochemistry, all the nitrogen cycling, all the sulfur cycling. And nope.
Oh.
It's not, I mean, we found like a couple of genes in
Huh.
viruses that might be tangentially related to this, but nothing in the main cycles. Like we know the genes in the main nitrogen cycles, the main sulfur cycles,
Mhm.
nothing. Instead, what we found was viruses were carrying genes that helped with oxygen stress.
Oh,
So of course
that's
they have
useful.
no use for oxygen stress. They don't care.
Right.
But it's, we
But the
think,
facultative anaerobes are the things that are kind of, yeah,
and these are
micro
most,
aerobic.
so, yeah, and these are
Yeah.
these genes were most found in viruses that were infecting bacteria at the oxycline, which is where the
Gotcha.
water goes from oxic to anoxic. So
Yeah.
that's the area we anticipate there would be the most oxygen stress on the host.
That's so cool. So just a quick review for people who haven't taken limnology or oceanography. There's a cline for everything, for every
Yes.
gradient in the ocean, the point where there's a rapid transition. So there's, so if there's a change in the salinity, there's like a chemocline or a halocline or something. And if there's a change in temperature really rapidly, there's a thermocline. Yeah. So for whatever you're studying, whatever kind of abiotic nutrient or whatever abiotic gradient you're studying, there's usually a cline for that.
Yep.
There's not a light cline, I guess. There's just the photic zone and the aphotic zone. People don't say like the photocline, right? Or do
No,
they?
yeah, they don't say that, but it's a term that hasn't been used for a couple of decades, and I completely forget.
That's fine. That's fine. The memory cline.
The memory cline.
It's when
If you
something's just on
haven't
the
used
tip
it
of
in a
your
while,
tongue.
it's
Yeah,
gone.
it's below the memory cline. Yeah.
Yeah, so considering, I mean, you're in an evolution class, a main thing, we've been thinking about with like, well, why don't the viruses have the genes for nitrogen and sulfur metabolism? And so something we have to consider is that unlike a cell, the virus has a capsid that is of a fixed size. The genome must fit into that capsid.
Right.
So they can't just keep accumulating genes and accumulating genes. There is harsh selection for only including the genes that will increase their fitness.
Yeah.
If the genes don't help increase their fitness, they're going to be eliminated. They're not going to be selected for.
Yeah, and also just in general, when you think about the evolution of anything, it's not always a matter of what the hypothetical perfect organism is. It's just what's evolvable within the constraints that are set, either architecturally or what happens to arise via mutation or whatever. So I think that is kind of a generalizable thing to other to living organisms too. Is that not everything will evolve just because we want it. Just because we can think of it doesn't mean it necessarily evolves. Although sometimes it does. Yeah.
Yeah, and there's genome size constraints with larger organisms as well.
For sure.
Yeah, genome size reduction. Maybe you've talked about that.
I have. Yeah.
Okay. With symbionts having smaller genome sizes. It's just that for the virus, it's that physical limitation. Their capsid can't just get bigger.
Except, are there some viruses that do have big genomes?
Oh yeah.
What's the deal with those Mimi and Mama
Don't
viruses?
make me say gyrus.
Oh, okay.
No, I'm kidding. I'm kidding.
That's just giant. Is that just giant virus?
Giant viruses.
Yeah, that just sounds kind of
giant viruses.
Yeah.
So yes, the largest viruses, I believe the largest one. Well, the first one was originally isolated from actually an amoeba. And these largest viruses can actually be larger than the smallest bacteria, larger genome. And those large so a couple of things about them. One is, they were what got started that debate on whether viruses are alive or not. They started it up again with the discovery of them, and there's a lot of people who want to put them on trees of life.
As kind of like a fourth domain or like a third domain or fourth domain
or even more the fourth, fifth, sixth. They want to put viruses on there, so that's where that got started. But also the super cool thing is that those viruses can be infected by other viruses.
Yeah, phages. It's
So it's a
insane.
Russian doll. You've got the
Yeah,
large virus infecting the amoeba, and then you have the satellite viruses
right.
infecting the large virus within that amoeba.
That's insane. Yeah, one of them's called Sputnik, right, or
Sputnik,
something. Is yeah.
yeah,
So yeah, there are viruses for viruses.
there are. So
That's
now that's when when I talk to my students in my
mhm.
marine environmental microbiology class, I asked them, I'm like, if you have a larger genome than some of the bacteria and you can be infected by viruses, are you alive now?
Yeah,
It's this very fuzzy line.
totally.
Because the students, up until they get to my class, they've been taught there are rules, right? Biology has lines. We define this there, we define it here. One of my students told me, they were like, living organisms do not read the biology textbooks.
That's a good line. Yeah.
Yeah, but I know what I was thinking. You brought up something on the perfect organism.
Oh yeah,
And
sure. Yeah.
I think I know the perfect organism.
What's the perfect
Um,
organism?
it's a virus that only has benefits to its host,
Oh,
or a virus that has where there's no effect upon the host. Um, and actually, my favorite example, it's not a perfect example, but there's this virus that can infect a cricket, and it changes the male cricket's song so that the male cricket is more attractive to the female cricket. And then when
and that's
those
the
two
only side effect of infection. It just makes
it's
you a better
not,
singer.
that's the problem. So
Oh
that would
no.
be
Yeah.
that would be mutualism,
Yeah,
and
that
see,
would be
the virus
beautiful.
causes the host behavior to change, so the virus gets spread, but it's
Yeah.
actually parasitism because it makes both the hosts sterile.
Oh, wow. I guess I do talk about in the class, or earlier on when I was talking about genome evolution and horizontal gene transfer, I did talk about sort of like a case of kind of there's a few wasps that have kind of domesticated viruses genomically that use them instead of venom, essentially to like
Mhm.
immobilize the
The
caterpillars.
caterpillars,
Yeah, yeah,
yeah.
yeah. So that's,
So
oh, sorry, no, go ahead.
go ahead.
Well, I mean that's a virus that no longer reproduces separately from the wasp, really. So it's only being copied from the wasp's genome, and then the wasp basically transcribes and translates
Mhm.
the viral proteins and injects it into the caterpillar, and that's kind of the fixed amount of viral particles that get into the caterpillar, but they still take down the caterpillar's defenses and make it a habitable place for the little wasp babies to develop. Um, so
So
not perfect from
yeah,
the caterpillar's perspective, but very nice
no.
from the wasp perspective.
So I know somebody who studies that,
Oh,
Galen
cool.
Burke,
Nice.
and I use that in my class, and I ask the students, is this still a virus? Because
Yeah.
the virus's genome is actually spread out throughout the wasp's genome in,
Mhm.
I forget how many segments, it's like 15, I think my brain is telling me.
I believe you.
And the virus genome doesn't even encode for the capsid genes. So I'm like, is this still a virus?
Right.
Or is it just now part of the host? And I'm trying to get students to understand how evolution happens, right?
Yeah.
Over time, these are just going to be part of the host.
Mhm. Is chloroplast still a cyanobacterium? Kind
Oh
of thing.
yeah. Oh, and then have you told them about the green sea slug?
Oh, that's contentious. That one's really tricky because people keep trying to. It's so hard to sequence those genomes without contamination, or you know what I mean. They're. I think that one's people have gone back and forth on that one. But yeah, talk about the green sea slug.
Oh,
I've been
yes.
staying
This is
away
a
from it just because I feel like I keep waiting for the dust to settle on what the genome
No,
actually.
I tell my
Yeah.
students all about, arguments that people are having in literature.
Yeah. Yeah.
And yeah, so the green sea slug will suck the chloroplasts
Hmm.
and basically all the contents out of the inside of this algae, and then they will embed those chloroplasts underneath its skin. So the slug can now photosynthesize technically. But the cool part, the even cooler part, is that now some of those chloroplast genes have been found within the slug genome. So are we watching evolution in real time where
Oh yeah.
the slug is now becoming a photosynthetic slug on its
Yeah.
own,
Yeah,
which is
absolutely.
cool.
That's so cool. Yeah, that's a pretty wild trick. So that there are, I think that one of the contentious things about that genome assembly was that it's so hard to just get slug tissue that has
Yeah.
no chloroplast tissue in it, that some people were worried that there was some problem with the genome assembly that was leading to that
Oh,
interpretation. But
I
I
haven't
haven't.
read further. They didn't get
Yeah,
a good assembly. It
I
was too
think.
fragmented.
Yeah, I think that was an argument that I remember hearing, but this might have been from a couple years ago, so I don't
Mm,
really, I haven't followed it super closely either. But I
yeah.
remember it's just such an exciting result that I wanted it to be true, but I don't I remember being like, oh no, like people are questioning this, so
Mhm.
I put it aside for a second. But yeah, those, I'm blanking out on the Latin name for the slug right now, but it's a very, very cool system. And if you Google photosynthetic slug,
Yeah, I mean,
it'll
that's
come
gonna
up.
come up.
It'll be the top hit for sure. Yeah, well, I don't want to take up too much of your morning. I know everyone's like super, super kind of laid out right now because of everything, the semester ending and everything. But thanks so much for taking a little bit of time to chat and catch up a little bit. This was really fun. Yeah, it
All
was
right.
great to hear about your research.
Yeah, it was great to see you.
Yeah. All right. Take it easy,
All right.
Jen.